So this morning I'm going to review some 21 years of amphibian research by my lab and our collaborators at Humboldt State, UC Berkeley, and UC Davis. Work that indicates that there's three different and distinct anthropogenic mechanisms contributing to amphibian population declines in Northern California. So these are artificial flow manipulations of rivers and streams. The second is introduced predators. And the third is forestry practices, which have impacted both terrestrial and aquatic amphibian assemblages. So I'm going to take these in that order. The first is a result of altering natural flow regimes, primarily by dams, to accommodate human needs for power generation, agriculture, flood control, and the like. Such alterations change the timing of important attributes of river flow dynamics, such as water velocity and depth profiles, such that the timing for key life history functions of resident animals, like the timing of reproduction, are compromised. These life history attributes have evolved over millennia to coincide with natural cycles, and when they're compromised, they can have devastating effects on population viability. Next slide. An example of such an effect is seen with the timing of breeding and egg deposition of the foothill yellow-legged frog on the Trinity River. On the top right of this image is an egg mass that has been effectively destroyed by stranding as water from an artificial flow receded. The opposite effect of flushing by artificial flow water is equally detrimental, and if it happens to a large number of egg masses, an entire annual cohort can be lost. Our work on the Trinity indicates that the frogs are probably keying in to the descending limb of the natural hydrograph in late spring to determine when to breed and deposit egg clutches. The red arrow at the bottom indicates the descending limb of the natural hydrograph. Unfortunately for the frogs, which are now rare along 40 miles of the Trinity below Lewiston Dam, the timing of their egg deposition coincides with artificially increased flow releases that are intended to flush hatchery fisheries, primarily fingerling salmonids, downstream. This results in the loss of most frog egg clutches along this 40-mile stretch of the river. The inset graph shows the natural hydrograph in light blue. The number of egg masses in 1991 is indicated by the orange bars. The purple line indicates the artificial release of water from Lewiston Dam. You can see the difference in the height of the orange bar before and after this release. Note that a second smaller release took out even more egg masses that were laid after the first release. Female frogs are capable of only a single egg mass annually, so it doesn't take too many years of this happening for there to be a dramatic effect on the population of frogs. The reduction of frog numbers is probably also affecting food web dynamics, especially amphibian-dependent predator species. Our surveys indicate that garter snakes are present in much lower numbers along this stretch compared with a comparable area along the undammed South Fork of the Trinity. We suspect altered flow regimes in other parts of the state may be contributing to the decline of this species throughout much of its range. Next slide. Next slide, please. Controlled releases affect other species, and we've also been working on the western pond turtle. I know this is an amphibian meeting, but this is an amphibious reptile, and Park is here, so figured it was appropriate just to at least mention this work. With the turtle, we found that cold temperatures are having a negative effect on growth. The release from the dams comes from the bottom of the lake, so it's much colder than what you would have at the surface, where turtles spend most of their time in the water. So And it also seems to be affecting their basking behavior. To compensate for the cold water, they spend a lot more time basking than a comparable population of turtles in the undammed South Fork. And the current flow regimes seem to be reducing habitat complexity through channelization of the river, which was once a dynamic, meandering river with a lot more habitat complexity. So this is ongoing work with the turtle. Next slide, please. So moving on to the second mechanism of amphibian declines that we've been investigating, introduced predators. Next slide. Evidence for this second mechanism comes from our research in the subalpine wilderness areas of the Klamath Mountains, where we've surveyed now over 700 sites across 3 wilderness areas. Amphibian declines here result from the introduction of non-native fishes into lentic habitats, primarily lakes, some ponds, in these wilderness areas, primarily for sport fishing. This is a common practice throughout the western U.S. Next slide. 2 of the 8 species that comprise the subalpine amphibian assemblage in the Klamath Mountains appear to be particularly adversely affected, the Cascade Frog and the long-toed salamander. Next slide. This pattern of negative impacts at ponds and lake sites by trout, as indicated here by the probability of detection, also holds for the Pacific tree frog. Note that all 3 of these species are highly palatable to trout in all of their life history stages. Next slide. The toxic species that are part of this assemblage appear to either be less or unimpacted, as the top graphic with the rough-skinned newt indicates, or they show a positive response to the trout presence, as in the lower image here with Bufo boreas, the western toad. The positive effect on Bufo boreas may be due to a lack of competition from other amphibians or to a reduction of invertebrate predators on toad tadpoles by the fishes. This needs further research. You'll hear more about our work in this subalpine ecosystem from talks by Karen Pope and Justin Garwood and Clara Wheeler, where they'll detail some of our latest revelations on the impacts of non-native trout on these subalpine systems. Next slide, please. The third mechanism of declines I'll discuss today is forestry practices. As I mentioned earlier, these impact 2 different species assemblages, one terrestrial, one aquatic. And I'll take the terrestrial first. Next slide. The first research project in which I was involved here at the Redwood Sciences Lab addressed the question of how do vertebrate species assemblages differ by forest Cyril stage. And this involved ornithologists, mammalogists, herpetologists. And we looked at a set of stands, young stands being from 30 to 99 years of age, mature stands from 100 to 200 years of age, and old growth stand county. And some of the strongest differences in numbers among forest age classes for vertebrates were found among certain bird species like the spotted owl and the marbled murrelet, and amphibians. For example, here we found significantly higher numbers of California slender salamanders in late Cyril forest compared with the young forest. Next slide. We also found significantly higher numbers of Insatina, the most common terrestrial salamander in the local region. The highest numbers in the late Cyril forest. Next slide. And we found significantly higher numbers of the Delnort salamander in the late Cyril forests. Next slide. In several subsequent studies, we've collected data on forest stand attributes at sites with and without a particular species in order to learn more about important habitat attributes. Depicted here is a recently derived habitat model using forest inventory analysis data. This model is for the Delnort salamander. And we actually tested an earlier model that Amy Lind and I had developed By modeling the forest attributes associated with both the presence and increased numbers of plethodonid salamander species, we have found that attributes such as high canopy closure, large down logs, understory hardwood component, and moist, stable, cool microclimates are important habitat components for these salamanders. Some of these key attributes are more likely to occur or be more fully developed in late Cyril forests, as they are part of the successional process that takes time to develop and are thus often absent or underdeveloped in younger stands. Next slide, please. And finally, on this terrestrial module, we've done a follow-up study, Amy Lind and Gary Fellers and I, and we followed up our earlier work by revisiting 36 of the stands that were sampled in the mid-80s, again in the mid-90s, approximately a decade later, with the question that we hope to test: could we find any downward trends across the decades with any of these terrestrial amphibians? Well, we did find some differences year to year, which are illustrated here for Insatina. We did not find downward trends across the 2 decades for the 5 species that we were able to analyze. The overall differences we found earlier in numbers between forest Cyril stage did hold across the 2 decades, suggesting that forest succession has an important influence on the composition of terrestrial salamander assemblages, both species and numbers. We know very little about the ecological role of these often very abundant animals in forest ecosystem processes, so it's not possible at this time to speculate on how these differences in species and numbers might be affecting these processes. However, we found no evidence to suggest that any of these species are currently declining. Next image. While the richness and evenness of terrestrial amphibians is clearly influenced by forest practices, overall there's no strong evidence that any of these species are declining toward extinction on any north coast landscapes. I can't say the same for several of the aquatic species that are being adversely impacted by forestry practices. Next image. In the earlier research that I described examining relationships with forest Cyril stage, we also sampled streams in these different age classes of forest, and we detected 2 aquatic amphibians, the tailed frog and the southern torrent salamander, occurring significantly more often at late Cyril sites compared with young and mature sites. It seemed unlikely to us that forest stand attributes related to Cyril stage were responsible for these differences, and much more likely that conditions in the streams themselves varied by age class and might be responsible. Next image. Next. There we go. A fortuitous research project courtesy of Caltrans, where a storm-induced roadbed failure dumped fine sediments into 5 pristine streams in the Prairie Creek Redwoods State Park, allowed us to explore the impacts of these sediments on native stream amphibians. These are the same kinds of fine sediments generated by unpaved forestry roads, a huge network of which dominate the landscape of the north coast. Next. Fortunately, this road failure left 5 streams in the park unimpacted by sediment, so we had controls with which to compare numbers. Here you can see the difference in sediments between the sets of streams as measured by pool bowl sediment depth and pool tail embeddedness. 10 pools were randomly chosen in each of these streams for these measurements. Next. 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 Who's asleep back there? Our study design here also included looking at differences in numbers of amphibians by stream habitat type, which I won't go into today, but to suffice to say the primary impact, fine sediments, significantly reduced the numbers of tailed frogs in the impacted streams. Next. It also reduced the numbers of southern torrent salamanders. Next. And it also reduced the numbers of the most common amphibian in our local streams, the Pacific or coastal now giant salamander. Next. More recently, we have investigated the question of what are the landscape scale effects of these anthropogenic disturbances on stream amphibians. These species are highly sedentary and specialized in terms of niche attributes, and therefore highly susceptible to isolation and fragmentation of their populations at the landscape scale. Sampling across the entire 790 square mile Mattole watershed and using the existing landscape mosaic to determine the representative sets of streams that we sampled, we found tailed frogs and southern torrent salamanders were present mostly, or in the case of tailed frogs, only in streams transecting late Cyril forest remnants. Next. In the Mattole, unlike Prairie Creek, sediments turned out to not be the driving stressor. When we compared stream attributes across this landscape, we found that water temperature regime showed much greater differences than any other variable we examined, and was the most likely cause of the absence or rarity of these two species in streams that traverse second growth or grassland. The two temperature profiles depicted here show the differences between the extremes, with the bottom profile that of a representative late Cyril stream and the upper profile that of a representative mixed second growth grassland stream. The two red lines indicate physiological thresholds above which these two species are effectively eliminated. This map of our sampling effort across the landscape, the open symbols indicate no detections. The association of these species with streams and late Cyril remnants has resulted in populations of these two species being fragmented and so widely isolated from one another that metapopulation dynamics are unlikely, and these species are likely on an extinction trajectory in this watershed. So I'm going to have to end it here, and I would like to go to the last image. Back up one. Sorry, back up two. There. It's clear from our work that both temperature and fine sediments are important stream attributes, and given the current status of forest practice rules as depicted in this table, it's also clear that we as a society need to work along the lines of protecting headwater systems in a better way. And I would conclude by saying that given the unyielding and uncompromising effects of gravity, better protection of our headwater amphibians and their habitats may be the single most significant thing we can do to recover our salmon and the vital industry they used to support. Next image. I wish to thank these people, and I thank you for your attention.